A very good afternoon, everyone. And we were supposed to start at 3. We are kind of 12 minutes late. Really sorry for the inconvenience. We are not very much sound with the technology, though um, we are with this technology for last couple of years, but still this technology is showing more power to us. So thank you so much for your patience. Myself, Sakshar Shaha, the assistant professor from the Department of Pharmaceutical Technology, JISU University, would like to welcome you all along with our eminent speaker of today, Dr. Chinmoy Patro, in this day 2 of faculty development program. We are very much happy to get such a great response from all the participants in this program organized by our department. Thank you so very much for your cooperation. Without wasting much time of yours, let me take the privilege to introduce our today's speaker. Firstly, I would like to thank him for his precious time to share with us, and I'm sure we are going to learn a lot of new things from him. So let me welcome warm-heartedly Dr. Chinmoy Patro. He's the scientist E of Research Institute, Developmental Biology, Pune, India. He has completed. Sir was a postdoctoral fellow at Max Planck Institute for Heart and Lungs Research in Germany. He has also performed postdoctoral thesis work under the guidance of Dr. F.B. Engel at the same institute in Germany. He has completed his master's degree from IIT Kharagpur in biotechnology and biomedical engineering and completed his bachelor's in Jadavpur University. If I talk about Sir's achievements, so this time would be very less for us to describe. So I would like to talk about few achievements of Sir. He has received JSPS Invitational Fellowship for Research in Japan for the year of 2019.
Still more you have to improve. Marks cut check
sorry for the. He has selected as Wellcome Trust or DBT India Aligned Intermediate Fellow for the year gap of 2019 to 2024. That means this fellowship is still on. He has selected as the head of the Max Planck Partner Group for the time gap of 2015-2010 in India. He has received Early Career Research Award from DST, Department of Science and Technology, in the year 2016 to 2019. He has awarded by Travel Award. He was the winner of the Travel Award in the Winston Conference 2012, which was held in Chicago, USA. He has secured, that's a proud thing to say for me, he has secured All India Rank 1, the first rank in Graduate Aptitude Test in Engineering in GET in 2005 in the pharmaceutical science. If I talk about his research interest, then he has a research interest towards heart regeneration, cardiac pulse development, and vertebrate genetics and others. It was very, very, very short introduction of Dr. Chinmoy Patro, our eminent speaker of today. We are really very happy and privileged, sir, to get you here amongst us. Our faculties of our department and various other departments, various other institutes are here to listen from you. Sir, if you kindly share your presentation and talk to us and elaborate your research findings.
Thank you very much. Such a, it's not a brief, it is a long introduction. And I'm really thankful to Dr. Maiti for inviting me to share our little bit research we'll share, and also I'll try to sell Zebrafish model that how we can use it. I hope you can see my slide in full screen mode. Yes. Hello.
Yes, sir.
Yeah, you can, and also you can hear me. Yes.
Yes,
I'm
yes,
audible. Huh?
we can hear you, sir.
I can't hear you actually.
Can you hear me?
I can hear you.
Okay, sir.
Can you hear me?
Yes, sir. Yes, sir. You're audible, sir. Perfectly.
Perfect. Good. So yeah, yes, always I am excited when I talk to pharmacists. Yeah, because I am a pharmacist. And so how to start? So I work on a Zebrafish as a model organism. So this is actually model organism, but Zebrafish can be used in different aspect of biomedical research. So why Zebrafish and why you do research? For all what we think about, the end is that we think about our pathological situation, and it can be very mild like hyperacidity, or it can be very severe like myocardial infarction or heart attack, or little bit of card, maybe spinal cord injury, or the vertebral disc degeneration, high blood pressure. So whatever the situation it is, if it is very strong or the mild, first thing that is what to do, we go to doctor, and then of course we go to pharmacist. And in this case, we get the drug medicine in the form of any kind of formulation, can be capsules or tablets or injectables, whatever, and we feel happy. So when you think about how it works, being a pharmacist, all of us know that, okay, this medicine, whatever is it inside the capsule or tablets or injectables, it's basically targets something. And this is the target is nothing that who is the culprit, who is creating problem. And if so, that's mean that if you want to discover a drug or medicine, we need to know that who is the culprit for that particular pathological situation. And if you know that, then we can find out, if we know the culprit, then we can find out how to stop that process and how to remove from that particular problem from the body. So to do so, we cannot work on humans. We have to find out animal models, and then of course they should have the similarity with human. And in that scenario, this is the reason that we use animal models in research. And if you talk to anybody, a layman, first, and if you ask about the animal in research, then definitely people will talk about mice, rabbits, guinea pigs, monkeys, and all about. But if you think that, okay, most abundantly used animal models are not belongs to this category. We mostly use, other vertebrates or lower vertebrates and vertebrates and invertebrates. So if we think about history of research, that more than 2,500 years before, people start to use sponge, because sponge has huge regenerative capacity. People use also Hydra, Planaria, C. elegans, because they are simpler model. They do have nervous system, they do have regenerative power. So we can understand the different neurological problem using this smaller animal models. Drosophila or fruit fly, they're very important model system. That today's in this medicine, what we are taking now, many medicine came because of the Drosophila, because we understood many signaling pathways or targets from this fruit fly studies. So fruit fly, more than 100 years, people are using, or scientists are using fruit fly as a model organism. And of course, we use the chicks, and among all these, youngest is a zebrafish. So last 3 decades or 30 years, we are heavily using this zebrafish as a model organism. So first, what I would like to tell you that zebrafish are basically our neighbor. So zebrafish mostly live in the eastern or northeast part of India, Nepal, Bhutan, and also Bangladesh, Myanmar. This area zebrafish are abundantly you can find in the rice field. Zebrafish are very popular, not because of the research. They are very popular because they are, the people use the aquarium. So they're the pet animal. And more than 100 years, they are roaming around the world because of the different, nowadays you can find different colors like this here. I'm showing that pink, green, yellow, and also green. All these beautiful, stripes you can see, and that's why it's very popular. And however, we are using only in the lab this gray zebrafish, these stripes. They do have this zebra-like stripe, that's why the zebrafish. So if you think about any kind of research, that basically ultimately we think about how human will be benefited from this. Whatever we are working today, how in a long term or short term goal, that how human will be benefited from this kind of research. If we look at the research different or using different model system, not the animal model, different model or experimental model, then the relatively simpler form is the cell culture system, because there we use only the cell. This is not the tissue-like structure, and we can observe under the microscope each and every cell behavior, cell migration, or how the cell division is happening, or the how mitochondria is working, mitochondrial fusion and fission. All these things we can basically observe under the microscope in case of the cell culture. However, this is not similar to the animal model. So whatever findings we are getting, basically we have to take it forward, that it's really in the at the level of the organs or animal level, this whatever our finding that can be validated or not. So from the cell culture, if we start, that goes to the tissue engineering, where we do have like scaffold like tissue. There cells are also there, and we do have the extracellular matrix, but of course in this situation we don't have like different organs. So that means that we have to again go to the next step, that is the animal model. And if we think about the animal model, then we mostly use the mice here, and then primates like monkeys, and then at last we all the clinical trial passed, then go to the human. So this is the process is very complex. It's like if you look at the road through the highway, this hilly area, you can see this is such a complex system. But one can make tunnel under this hilly area, and it can shorten maybe 80-kilometer road, you can make like 8-kilometer tunnel. So what I compare here, that zebrafish is like making tunnel. So whatever we are seeing here, that all these processes you can basically do it using zebrafish as a model system. Like whatever we are seeing, that all these processes we can also see using zebrafish model. And also, of course, it is a animal model. So why I'm telling that this is a linking, or we can do almost everything what we are doing with the cell culture, and also what we are learning from the mouse, that we can do using only zebrafish. So because that their fertilization in vitro. So here I am showing you that this, you can see that yolk sac, this is basically fertilized cell. So I'm showing you a video. Here I kept one male and female together and in a breeding tank, and you can see that this, the mustard seed like particles are dropping here. So they're basically nothing. They're the fertilized eggs. This female throwing the, the eggs, and the male throwing the sperm, and fertilization happening in vitro within a millisecond, and basically they're going down. If you look under the microscope, they will be looking like that. On the top, you can see the yolk sac is written. Yolk is there and fertilized one cell, and within 30 hours, you can see the developed embryos. Here you can see this, green color. That is basically I'm showing the blood vessels and live zebrafish, because they are very transparent. So we can see every cell types, every organs, how they are growing. So here I'm showing the blood vessels. So basically we can observe the growth, animal growth, or development of an organs, or maybe cell division. All these things one can observe this like in vitro cell culture system. One female can lay hundreds of eggs, so you can get a lot of fertilized animals or eggs to study, their different kind of drug screening, or maybe you want to know the molecular function, and everything can be doable. Nowadays, with the help of CRISPR-Cas technology and also other techniques, we can easily make knockout and also transgenic animals to see different organ development. So of course, these are all the advantages, but then again raise question that, okay, how much similar with human? Because if there is no similarity or very minimal similarity, then no use. So if you look at the genetics level, so that you can see that more than 12,700 genes out of 20,000. So around 70% genes are common, and they do have all the organs like we do have, except lungs. So that means the function of all these common genes, 13,000 genes, can be studied using zebrafish as a model organism. So now again, so that, okay, since we work on heart development or regeneration, I am going to give an example that what we can do with this model system. So this is just 2 days old zebrafish embryos. You can see the yolk sac, that is nothing, it's food reservoir. And this, this red box is showing the pericardial cavity where heart is residing inside, the pericardial cavity. They do have 2 chamber heart, not like us, 4 chambers. So here you can see this green heart with 1 ventricle and 1 atrium. And this is from a transgenic animal. You can see all cardiomyocytes, the only contractile cells. So you can see the shape of the cells, each cardiomyocytes. If you use double transgenic, here I am using the red and green. Green basically showing the cardiomyocytes and red is showing inner endocardial or endocardial cells. And in between the gap, you can see this is basically cardiac jelly, which give the frictionless movement of the heart. So if you look at the 2 days old heart, here they have the ventricle and atrium, and in between they do have the atrioventricular canal. This arrow is showing this, but they do not have valve at this stage. But if you wait another 7 to 8 days, then you can see that they do have nicely formed valve leaflet at the atrioventricular canal, and also you can see that very simpler structure of the ventricular wall, it became very complex. So they do have like finger-like projection inside the ventricular lumen. It's called trabeculory. Similar like the mammalian system, they do have the trabeculory. They're very important. So is all the images I have taken from a living animals. That is the most important. You don't need to kill the animal. You don't need to manipulate this animal, but you can see each and every event is happening. So if you look over the time, so one can really see that how the valve is developing and how the blood is flowing. Everything can be visualized in a living animal. So it's using a very, very simple microscope. What I'm saying, and this video has been taken with the help of our mobile phone through a microscope. So you can see that the heart is beating. This is a 4 days old heart. You can see the blood is coming to the atrium from the bottom, go to the ventricle pumping the blood through the outflow tract, OFT outflow tract. So and then it's going toward the body. If you want to use the fancy microscope and you want to know more in details, like at the cellular level, so you can also use the light sheet microscope. So here I am showing here that the red dots is basically the nucleus of the cardiomyocytes, and the gray, whatever white color you are seeing here, that is nothing, is filamentous acting in a cell. So each and every cell you can see during contraction and relaxation how much expanding and how much coming back. So one can really see the the force one can calculate using this movies in a living organism. So of course we need to know that what is the history, how this zebrafish came into research. So in around 1965, 1970s, George Steininger, he actually was a fish lover. He had the pet, the zebrafish in his house, aquarium in his house, and he used to observe every day that how they are behaving, and he found interest. So he started to do the research at University of Oregon in late 1970s, and then he published a couple of papers. However, in 1984, due to sudden heart attack, we lost him. Fortunately, 4 of his colleagues continued his zebrafish research in University of Oregon. So it is very slow movement. If you look at the publication in 1973 until 1996, you can see very minimal number of publications coming out. So means only the 4, 5, 6 people throughout the globe were working using zebrafish as a model. However, in 1990s, Christian Olson followed from Max Planck Institute Tübingen, along with 4 other the groups in United States. They have started to find out that how the existence of the similarity between human and zebrafish genomes or genetic similarity. And then there are 6 years of work of all 5, 6 groups. They have identified that hundreds of genes having the similar functions between human and zebrafish. And that is in 1996, they published around 50 papers. And from that day, as soon as they discovered that the zebrafish genetically is very similar to the human, and then many people start to working on the fish. And now you can see that around in 2020, around 4,500 publications in PubMed, and now more than 5,000 every year publications are coming the work on zebrafish. And at present, more than 500 labs throughout the globe, we are working on zebrafish or using a zebrafish as a model. if we look at the area we work, that one can do the modeling of human disease. I will give you an example. One can do the potential drug screening, toxicity study, and of course regenerative medicine, because they have huge, capacity, to regenerate their damaged organ. First is, the modeling of human disease. What is the modeling of human disease? So now, because of the sequencing, the power of the sequencing, we know that for different genetic disorders, that we know that which genes is basically problem. So, child born, live births happen with some, problem, and then one can find out that what genetic alteration happens. So what one can do, one can make the zebrafish that having the similar kind of mutation or similar kind of genetic issues. Since the genetic function are almost similar, so basically we can see the similar phenotype. So like for this case, like is the gene or TBX5 mutant, so you can see the four-limb knife did not develop properly. In case of the zebrafish, and also this poor kid having the problem with this, the heart development problem and all these things. But the zebrafish mutant, also this TBX5, you can see the spectral fin that is just like our, the forehead, our hands actually. And they don't have this spectral fin, you can see, and they do have the problem with the cardiac problem. So similar phenotype comes. So what one can do, one can take this zebrafish, the mutant having the similar phenotype and screen for drugs, because now there are, lakhs of FDP molecules are there, but we don't know for many of the cases their function. So one can take these FDP drugs and screen for the molecules randomly, and one may find out that okay, one molecule maybe replace the function of this TBX5 gene. So that way, basically personalized medicine, come out. So nowadays is very popular. So you basically make the similar mutation or similar genetic alteration. What basically you have the patient already having it, and you make the zebrafish mutant, and now screen for this lakhs of the molecule to identify which is useful. And I can tell you it is time consuming, but it's not so time consuming when you use the zebrafish. That I will also explain later on. Next is the cancer. So if you know that okay, this genetic mutation is particularly lead to the cancer, so you can make the model also for the drug screening purpose, or to understand the cellular and molecular behavior in the case of the animal having that mutation. So there is known already this in a human RAS gene mutation basically lead to that melanocyte carcinoma. So what scientists have done, that basically they have generated a transgenic line where basically melanocyte specific promoter is there, and which express this human gene, this mutated form or cancer causing gene now. So when you do this, you can see the control, and in case of this the animal, transgenic animal which expressing this cancer causing human gene, basically they have the black caudal fin, and then also you can see the hyperpigmented caudal fin in case of the C and also D. So you have the model system, and now one can again that the same molecules what I talked about, the FDP molecule, one can screen for that which molecules express this situation. So for small molecule screening, that of course that first thing that okay, being a pharmacist, one definitely think that target has to be conserved, because if you find a molecule which basically giving nice effect on the zebrafish, but it does not have any role in case of the human because the targets are not similar. So what we found that only more than 80% targets are similar. So it's pretty high, conserved targets. Drug metabolism, that is also important. I don't need to explain in this audience. So they do have the similar drug metabolism like human. If you look at the pharmacology, also pretty conserved, and the physiology. If you look at the ECG, you can see the mouse ECG, you can see the zebrafish ECG, and the human ECG. So zebrafish ECG is much closer to human than the the mouse, because mouse heart beats around 500 to 600 beats per minute. So cardiac frequency much higher, more than 5-6 times higher than the human. So that if you look at the cardiac physiology, that zebrafish is much closer to human. So how we do it? So this, in case of the embryonic screening, basically we start for every screening, we mostly start with the embryo. So you can use the 96 plate format, and every well you can put 4-5 embryos. And you can see 96 plate format means you can have the 2 control probably, and then 9 to 4. So one screening, 94 small molecule you can screen at a time. So that's why it is high throughput screening, and this way you can do, find out that any molecules which can be useful for any disease conditions or a toxicity study, also behavioral study, anything can be doable using this 96 well platform. Now come to the regenerative medicine. So basically, we need to study this, the regenerative medicine or organ regeneration. If you think about that organ injury, it can be drug induced, it can be genetic, anything. If you have any organ injury, basically what we can do, we can't regenerate. So human cannot, most of the organ except liver, we can't regenerate. So what you need to do, you have injured organ or damaged organ, we basically try to replace it with the donor organ. But getting a donor organ is not so easy. So the demand is much higher than the supply. So what you need to do, you can, that whatever we have, that little bit of like maybe 30% damaged organ, but we have 70% organ is still functional. So one can induce the regenerative process. That is the one. And another one can be that you engineer this, the functional tissue outside, and you can just basically replace this damaged part of that particular organ with the new tissue. But whatever you want to do, whether you want to induce the organ regeneration or you want to basically engineer new functional tissue and then replace it, in both the scenario, we have to basically, what you have to do is to understand that how one can induce tissue regeneration. And that understanding can be done only for the animals naturally available that who are naturally, regenerate their organ. So you can again come back to the animal models which can regenerate their damaged organ. So again, I'll go to the history. If, as I mentioned, the spawn, similarly, like also in 1740, this Italian scientist Abraham Tremblay, he has shown this hydra. Basically, if you cut into two pieces, there's two hydra they basically generate over the time. In case of the planaria or this flatworm, if you is more than 200 years ago, I've shown that if you cut into 279 pieces, one planaria, 279 planaria basically form from one planaria. And I think many of us know that, okay, sea star, if you are starfish, if you cut one limb of, they can regenerate that new limb. So they have tremendous regenerative capacity. So this is a very nice module to study the, in general regeneration. When you come to the organ specific regeneration, of course, then you come to the artifacts. Basically, that 350 years back, BC, Aristotle described that this house lizard, they can regenerate their tail. I think all of us experienced with this during our childhood, that if they touch the tail of this house lizard, this tail will fall and they can again regenerate. So, more than 2,500 years back, Aristotle described this scenario that, okay, this house lizard can regenerate their tail. In India also, many labs nowadays also studying this tail regeneration phenomena using house lizard. Very recently, in the last decades, we found that mouse and also like the human, we can regenerate our the heart just immediately after birth, but it's basically lost our regenerative capacity as we grow up. But in other organisms like axolotls, newts, they can regenerate that many organs, and tadpoles can regenerate their limbs, but not the frog. Frog can't. But adult zebrafish, even like 1 year or 2 years old zebrafish, they can regenerate almost each organ: heart, fin, spinal cord, cornea, cornea, pancreas, everything they can regenerate. So it's like a beautiful model to use to understand the regenerative biology. And another beautiful thing is that their regenerative time is very short. Within, like, for the heart, if you make an injury within 2 months, they can complete their regenerative process. Of course, when you start to study the regeneration and you understand the regeneration, that you have to make the model, that you have to make the injured model that for that purpose. So like all of us know, yeah, that gentamicin is basically kidney toxic and acetaminophen is basically liver toxic. So if you feed this fish with the gentamicin, then you can get the kidney injury, and then one can study the kidney regeneration. Same thing with the help of the acetaminophen high dose, we can make the liver injury and then study the liver regeneration. Similarly, we can do the genetic, like genetically we make the organism and very specific, the organs can be, make the injury can be induced in a very specific organs like the heart or pancreas using the different toxins like diphtheria toxins A or maybe the enzyme nitroreductase, which basically an enzyme that convert the metronidazole to a toxic substance. So if you have this transgenic animal and you feed with a metronidazole, basically that particular organs where nitroreductase is expressing, those cells will die. Similarly, surgical procedure of course we use heavily like dissection and cryo. So for example, in case of the amputation model, here what I am showing here, the caudal fin regeneration in chicken. This is the caudal fin or back fin. You can see uncut, and after the cut, 1 DPA means 1 day post amputation. And you can see that after 3 days, 6 days, how it is growing, and within 20 days, like in 3 weeks, they can completely have the regenerated caudal fin. Similar also the thing is that the heart, if you make the injury within 2 months, they can have the fully regenerated the heart in case of the amputation model. In case of the cryo-injury model, it is nothing, it is a cold burn. So you can touch a very cold probe like copper probe on the tip of the heart, and you can burn a specific like 25 to 30% of the tissue, and you can see that the regeneration. I'm going to show you that. So my lab is actually interested to understand the heart regeneration and the heart development or valve development. This is the major area. However, we work also like intervertebral disc, spinal cord regeneration. So many areas we are working, but our main focus is the cardiovascular biology. And these are the people who work. So I should make a that virtual tour also here, that we keep the Zebrafish in a system. This is at Agartala Research Institute. We maintain it. And every tank having a different kind of fish. We generate a lot of knockout and transgenic animals. That's why we need to use different tanks to keep different knockout and transgenic animals. They're just 2.5 to 3 centimeters long, not more than that. You can see here the adult fish I'm showing here. And the left side and the right side, you can see is very tiny animals. They're just 2 weeks old. You can see that these very tiny larvae, they are moving around. Right side and left side is the adult animal. So yeah, so these are the system. The water quality basically maintained by a central unit, which maintain the temperature, salinity, and of course there should not be any toxic substance. So filtration and everything. And here I'm going to show you the breeding tank. I have already shown a video before. So this is the breeding tank where we keep basically male and female. And like if you want to have tomorrow morning eggs, we can keep it tonight, and tomorrow basically we can have hundreds of eggs to do whatever you want to do, like micro injection or drug screening, whatever. So why we are suddenly interested to the heart regeneration? Because I have started heart regeneration study after coming back to India. Before, during my PhD and postdoc, I was working on like valve development, trabeculation, all these things. But my interest was that if I start my own lab, I should look on the heart regeneration, because if you look at that, global cardiac cases, then more than 15 million people die every year due to the myocardial infarction of heart attack. So you can see left side, this is I'm showing here mouse healthy heart, and the right side you can see the infarct written there. This basically we blocked the coronary vessels. On the bottom you can see the coronary vessels in green, and red are indicating the block is. If you make the block, the circle area is showing the tissue, cardiac tissue, which basically gets the blood supply through these blood vessels indicated by area. So if you basically block this blood vessel or tie it with a suture, then basically you get that the tissue having no blood supply or lack of oxygen or the food or nutrients. So lead to the cardiac muscle death. And including human, adult mammals cannot regenerate their heart. Means we have very, very limited regenerative capacity. So what happened, we can see here the sections I'm showing here, the blue basically here the scar tissue. And all of us know that okay, there is a cut mark, that is also scar tissue. This scar happened, and then here you can see the really healthy muscle, that is the very great, or you can see. So, this that means in one attack, around 25% to 30% of the cardiac tissue basically get, completely die. And since these cardiomyocytes cannot, repopulate or new cardiomyocytes, does not come, basically this area remains like scar tissue, and that is non-contractile. So that you imagine that every, like if you have 3 attacks, so that means around 75% to 90% that's cardiac muscle damage. So the person does not have any scope to survive because heart rate becomes a non-functional. So we cannot do it. But if you look at the zebrafish, if you make the injury, they do have the scarring, but they can resolve this scar. The scar can be, replaced by the newly formed cardiomyocytes. So you can see here. So zebrafish having this tremendous capacity to do this. And what we want try to understand, we want to learn, or zebrafish basically is our teacher to teach us how they can regenerate their heart, and we want to understand. So what we do for that, I am going to show again in a video. So we basically get first by each. So this Ganesh is showing you that how to make it. You put in an anesthetizing solution, so the fish will fall asleep within a minute. These are the very simple instrument what you are seeing here. And he's putting this copper wire in liquid like nitrogen, so it will be super cold. And then he is keeping this sleeping fish on wet sponge, keeping the thorax upward. Now he is actually removing the scales. So he'll remove the scales and then try to make a small hole through which basically heart will pop. So you can see now that heart is coming out. Yeah, I hope that everyone seeing that. That's a beating heart. This is ventricle actually. This ventricle actually popping up, and he will now touch this super cold cryoprobe on top of it. Yeah, this way he basically making injury 25 to 30% of the cardiac or 35% of the cardiac. Everything done, no bleeding, nothing. So now the heart inside and then everything. So you put into fresh water. So now it start to, gill movement has started. So you see now that it will start to fail, and basically it is moving, so it's done. So now he will keep it in the system. So what to do? I'm going to show you that. So now is the transgenic, that red cells you are seeing basically this is the cardiomyocyte injury. So when you make the injury, you can see that most of this the black area that leaves is very few cells. So that is injured area. And over the time, basically this area get covered with the cardiomyocytes. So his very talented student, Devanjan Mukherjee, he has started this work. And what he has done, he has screened for the molecules that which basically would upregulated or induced in case of the injured heart, because we know that zebrafish can regenerate their heart. So we decided that okay, let's find out that okay, which molecules are goes up. So probably they help in heart regeneration. So he found there are also many molecules found, but his interest was like CTGFA and CTGFB. These molecules is almost 4 to 6 fold up regulated. So he said, okay, I want to work on that molecule. I'm not going to show all the data. I'm going to show is very limited amount of the data. So CTGFA basically a secreted molecule is basically produced in a cell and secret out. So what we have seen that it basically secret in the produced in the endothelial cells or endocardial cells and goes into the extracellular matrix. And if you when you compare, they are very similar with the zebrafish CTGFA is very similar with the human CTGFA. So that means we think that okay, these functionally they are the similar or conjunct. So what Devanjan told that okay, let's see, want to know the function of this gene, so make a knockout of fish. So then he made the knockout zebrafish with the help of that CRISPR-Cas technology, and then he said okay, compare the regeneration between the wild type and this knockout. So this what he has done, he made the injury, and after 60 days or 2 months, he basically looked for the regeneration. So upper panel, what you are seeing, this is basically wild type scenario that we know that the heart can regenerate, and bottom in the mutant or knockout, CTGFA knockout. So in these animals, CTGFA is not there. So you can see this boxes basically showing the injured area. This yellow boxes showing the injured area. You can see the upper panel. And if you look at that, basically that green is the cells and the red is the cardiomyocyte. So you can see that this injured area all fully red cells are there, so indicating that they are fully regenerated, the red one. And the orange box basically showing the healthy region that should be remain always healthy, and you can see it's also present in case of the wild type scenario. What happened in case of the mutant, if you see, if you look at the yellow square in the mutant, or where CTGFA minus minus, you can see there's a huge area. There only the DAPI means blue means the nucleus, but there is no red cells. So that means there is no cardiomyocytes. So they fail to regenerate. However, if you look at the healthy area, that is the orange box, they're quite fine. So that means that the rest of the heart is fine in the mutant, but the problem is the regeneration. So that means they can't regenerate. CTGFA is required for the regeneration. Then he removed the heart and look at the heartbeat. And what he found that, okay, now you can see this wild type, and you can see here also mutant, that mutant heart is, this is also very huge scar tissue. So that means, they fail to regenerate and the heart is not functional enough to circulate the blood. Then again, he looked at this, again, the scar. So you can see that wild type scenario, left side, CTGFA++, you can see nicely regenerated, but in case of the CTGF plus minus, that is head, and minus minus or the mutant or knockout, you can see they have a lot of blue. So that means they fail to regenerate. Then he asked, okay, if we give extra CTGF, can we induce the regeneration? Can we make the faster regeneration? So he has first looked at, okay, what happened with the cardiomyocytes and the blood vessels. So here, what I am showing here, that the red is again cardiomyocyte nucleus and green is the blood vessel. So very interestingly, what you observed here, that you see that after 4 days, that few cardiomyocytes in the black region in tissue, they migrated. And if you look at the double, like green and red together, then you see this, all the red cells are basically overlapping with the, this coronary vessels or green vessels, blood vessels. So that means that cardiomyocyte migrate slowly into the injured tissue through the blood vessel. But in case of the mutant, you can see right panel that there is a lot of blood vessels, but not a single cardiomyocyte. You can see that black area is completely empty, no cardiomyocyte. So that means that CTGF required for the cardiomyocyte migration, and also the migration happen along the blood. Here you can see, if you look after 12 days of injury, that 12 DP7 means 12 days after injury, you can see in case of the wild type, that is CTGFA++, you see a lot of cells already inside the injured tissue. However, the head, that is the CTGFA plus minus, or the knockout, CTGFA minus minus, you can see very few cells, almost nothing, no migration, as I have shown on the upper panel. So next, he just basically look for the regeneration induction. So here, generally, what we have used, basically used a reporter means overexpression transmit line, if that express CTGFA extra amount, if you give the heat shock. We normally keep zebrafish at 28 degrees centigrade temperature, but in this animal, if you keep them at 37 degrees for some time, they basically express extra amount of CTGFA. So what you observe in case of the wild type, you can see there are relatively less number of cell proliferation, but you can see here also the after 30 days scar is still there. But in case of extra amount of CTGF, that is the HSP70 CTGFA, you can see the blue area almost invaded by this the cardiomyocyte, that is a brick red. So that means if you give extra amount of CTGF, you can induce regeneration. So altogether, what we have, Devanjan has shown that CTGF is important for that regeneration, and if you give extra CTGF, you can promote the regenerative capacity. So this is actually I am not going to discuss all this detail. So this is the finding and also is published last year. With this, I would like to thank all the collaborator and all the funding agency, and thank you all for listening me. And again, I would like to thank Dr. Maiti for inviting me to share my our research work. And I'll be happy to discuss if you have any questions, please ask.
Thank you so much, sir. Thank you very much, because our own lab in JS University, a few of the faculty members are also doing research on zebrafish. We are trying to develop few models, and hoping the faculties who are involved in doing that have got a lot of information. I myself have got a lot of information, because we're doing research on living animals like rat or rabbit requires the ethical clearance where we can do zebrafish model in a lot of studies, and where to handle them, though it is a little difficult as they're very small, but still there's no hectic thing like getting ethical clearance is not there. So thanks a lot to share about the zebrafish model and how um, applying various genes, like you were explaining CTGFA, how it helps to regenerate the cardiomyocyte and helps in regeneration of the heart. Thanks a lot, sir. So I would like to request all the participants, all the faculties from different colleges and universities who are present over here. Your platform is open for the question and answer. The questions you have, the kind of questions you have for your researcher regarding zebrafish, regarding today's session, kindly put the questions in the chat box. I'll read out for sir, and I'm sure sir would be very happy to answer that. And those who are watching us from the YouTube, because the session is also live streaming in the YouTube, you can put your question in the YouTube as well, so we can take the question from there. Uh, we have already a question in the YouTube, sir. Just a moment. Uh, our own faculty member has raised a question, sir. He's Victor Roy Chaudhary. So he has said that the heart of zebrafish has got 2 chambers while the humans have 4. How can we relate the cardiac function while any preclinical studies?
Yeah, so you know that you have 4 chambers is because we our the blood going to the lungs and coming back. But if you look at the functions like the ventricle and atrium, that is not different. That is the same process like SNO sending the signal and coming to the ventricular apex, and as it happening in the mammalian system. But only the limitation is that if you want to look for the ventricular septum formation or septal defect, that cannot be studied using the zebrafish because they don't have it. But the rest of the function and data, whatever you want to do, as I shown that if you look at the ECG, it's pretty similar, and their heart also like adult zebrafish heartbeat like around 80 to 90 beats per minute. So if you look at the ECG, if you look at the the cardiomyocyte structure and functions, they are pretty similar. Yeah.
Thank you, sir. Sir, there is another question from another faculty named Abhishek Jana. So he's asking that, is there any chance of using tank breed against wild type, as nowadays some researchers are going for tank breed, and how much different they are?
Tank breed, I did not get actually. What does it mean? What you mean here?
Abhishek, if you can specify, or I'm just Abhishek, you can unmute yourself and also ask or clarify the question.
Uh, Good evening, everyone. Generally, my question is about as we know, tank breed. I'm saying the different color fishes, like different from the wild type. Wild type is generally the silver color with the black stripes. Nowadays, I have seen some people are actually using the different type, like they have different color options like that. So they are going for that. So how much different they are, and can we also study on the tank breed?
No, actually, you know that it depends what kind of study you are going to do. If you want to study functions of a specific gene and or the pathways, anything that we always go for is inbreeding. So we know that okay, these genetically they are similar. It's not like making outcross with that different color or different stripes pattern, that we actually don't do. So yeah, but if somebody wants to study that, okay, that how if I cross like this pink fish with the gray fish, and then try that whether any this outcross having any effect on anything, can be like body stripe pattern or color pattern, then it's fine.
Thank you, sir. Sir, one question I have from myself, sir. If we are going for drugging, like if we have to if you have to incorporate a drug to zebrafish, And in animal study, we used to use some specific dose, but here, how do we can, we can give a specific dose to a zebrafish because
Yeah, good.
we cannot
Yeah,
give it
so
through oral route.
I can explain you this one.
Yeah.
So actually what we do, we always look at that what is, if you known drug, what we are using. So if you know that okay, the mouse model we use to reach the blood concentration like, 5 mg per ml or whatever. So what we do in case of the zebrafish, you don't need to inject or feed because zebrafish they are very much permeable. So the embryo water, we make this concentration that 5 mg per or per ml of water, embryo water, where your embryo is there. So basically, you make keeping the similar concentration when you are treating for the treating the embryos. So if you want to go for the drug screen, then of course you don't have an option that you don't know the dose. So what you need to know that you have to find out that what is the dose basically, not maximum dose, basically not killing the animal or not making any deformities. So if you know that okay, this dose, this animal is fine, maximum dose, so suppose 20 microgram per liter or whatever, then you can go like 20 microgram per liter, 15 microgram per liter, 10 microgram per liter, 5 microgram per liter. So like make the 5 dose or 4 dose and then look that okay, the pharmacological effect.
Okay, so we have to make the concentration of that particular concentration in the embryo water, and that's we can incorporate the drug.
Yeah, so you consider like our blood concentration is similar to the embryo water.
So there's no oral feeding or something like that, that we cannot, as they're very tiny.
No, we can not even tiny because oral feeding you cannot control, because in case of the mouse you can control, but here you cannot control, and that's why we do in case of the adult. So of course, when you make the first screening, then you have to make the secondary screening, and this secondary screening has to be on the adult because that okay, that metabolism system is different in the embryo and the adult. So when you go to the adult, we basically make the intraperitoneal injection.
Sir, one more question, if we are giving, let's say 5 microgram per ml, we are making the concentration, we are putting it, or we are making the concentration of the embryo water, that is it very much assured that that much concentration will be inside the embryo and that will be in the body of the fish ultimately,
yeah, it is more or less will be remain like
same.
Yeah, it is remain like this, but of course you cannot keep it for 5 days. You need to like change every day.
Okay. And then another question, I think somewhere in your slide we have seen that you are doing kidney injury by using the gentamicin. So if we want to do some injury by chemical induced injury in gentamicin, how, or any other drug or any other chemical, so how do you gonna do that?
This all whatever the drug administered, you can also administer in the water, because this like go through the gills and gills will absorb.
Okay.
So you can throw in the water, and otherwise if it is water soluble drug, you can throw in the water, but then its amount will be more. Otherwise you can inject intraperitoneal, that you can slow release, and also you can inject. We have done angiotensin injection last year, so you can do it like intradermal also. Intradermal, intramuscular, all are possible.
And sir, instead of putting the drug in the embryo water, as you were saying, if we put the drug in the adult fish, the watered adult fish are moving around. If we put the drug there, will it give the same effect, or it will be a less effect than embryo water?
No, as I told, this is two different scenario. The adult having different metabolism system.
Okay.
And embryo having the
Different.
poor metabolism system, because they are still liver to grow, pancreas to grow. Their metabolism system. So probably that in case of the embryo, you will see that lower dose giving the effect. Maybe in case of the adult, you need a much higher dose. So those things have to be. But people use the embryo because then you can do high throughput. If you want to take the adult, then you cannot do the high throughput. So high throughput first. If you want to do the drug screening, but if you have any specific like 10 molecules, definitely you can directly go for the adult.
Okay, sir. Okay. Thank you so much. So there's another question from Abhishek Jana again. He's asking that what can be the best procedure to dissect zebrafish without harming any internal organ?
You can open the chest and abdomen. Simply cut, like,
Right.
yeah, that you can do, and you will not harm any internal organs. So everything will be visible. And then maybe one side you can cut with a thin, scissor. One side, then you can see each and everything, like liver, pancreas, intestine, everything.
Let me see if we have any other question in the YouTube note. Okay. Not yet. So I hope, sir, you have answered very explanatory all the questions of us, and thank you so much. We have few questions, we have few confusion about it, about zebrafish research, and thanks a lot, sir. We have for you a very small token of appreciation for sharing your time. With your permission, I would like to share that on the screen, and our coordinators will definitely communicate the same with you through email. This is a very small token of appreciation for you, sir, for sharing your valuable time and
Thank you.
your knowledge. Thank you so much, sir. Thank you, sir, for being with us. Thanks a lot, sir.
I hope you enjoyed it.
Absolutely, sir. Absolutely.
May I leave?
Sure, sir. Sure, sir. Sure. Now, I would like to hand over the rest of the part of the program to my fellow colleague, Dr. Debjani Sarkar.
না এটা তো আমার ফটো আমি কি জানি কি লিখে আইছে এখন মিউট হয়ে গেছে কেন আমি তো ফোন নিয়ে আসব না নাকি
Please mute yourself. Please mute yourself. Okay, so very good afternoon to all of you, all the participants and our delegates. And I would like to welcome you all in our second day FDP and second session. So in our FDP, next speaker, honorable speaker is Professor Dr. Umesh Kumar Patil, MPharm PhD, Professor Division of Pharmacognosy and Natural Product Research, Department of Pharmaceutical Sciences, Dr. Horishin Gaur Vishwavidyalaya, a Central University, MP, India. So I would like to introduce our speaker. Professor Umesh Patil has been working on ethnopharmacology and evidence-based medicines for last 20 years. Presently, he is working as Professor of Pharmacognosy and serving as General Secretary to Indian Society of Pharmacognosy. He has also been serving as board member and vice president of International Council for Medicinal Plant Research and regional representative of ICMAP for SAARC countries. His area of specialization is ethnopharmacology and herbal drug technology and natural products. He has more than 22 years of experience in teaching and research. He is recipient of 10 prestigious awards given in the field of HMPS and remained Boy Scout Fellow of DST, Government of India. He has been associated with IAC for last 15 years, and he is a member of many professional bodies, including International Pharmaceutical Federation, International Society of Ethnopharmacology, International Society for Medicinal Plant Research, Germany, International Pharmaceutical Student Federation, International Society of Natural Product Development, APTI, IPA, IPGA, ISTE, IASTAM, and IAHP. He has published 112 papers in reputed journals and has been serving as editorial board member of many reputed journals. Professor Patil has done various academic visits in different countries, including Spain, Switzerland, Netherlands, Belgium, Turkey, South Africa, Germany, France, Thailand, Sri Lanka, Malaysia, US, Australia, etc., for scientific assignments related to ethnobiology studies. Welcome you, sir, in our JIS University Pharmaceutical Technology Department.
Thank you, madam. Thank you so much for your nice words in my introduction. Yeah, is it audible, ma'am?
Yes, it's perfectly audible, sir.
And also, I'm grateful to VI University for giving this opportunity to speak with you today, especially on the scope and the challenges which are associated with natural products and the products of medicinal importance which are come from the natural sources, specifically from botanicals. So I'm grateful to the university and the Arinda Mati and the joint coordinator Ritu Khandra and joint coordinator SK Abdul Amin for their kind invitation extended for giving this particular lecture. So my topic is, just a moment, first of all, I will share my presentation. Just a moment. So, is it visible, my slides now?
Yes, sir. It is visible.
Yeah. Thank you. So I'll be talking about what kind of challenges which are associated with the natural products and the products of medicinal importance, which are either coming in terms of active fraction or sometimes in terms of the lead compounds. So these challenges are many times we must consider as an opportunity for the medicament or novel drug delivery system. So since we have different kind of diversity of the healthcare processes which we involve in our country, and also we have placed with the very good well established kind of traditional medication system. And we know that this particular medication system of our country is dependent on natural products. Most of the natural products, they are isolated, or they are evolved from the botanicals. So in what way we have to consider various kind of challenges in terms of phytomedicinal research. More specifically, we can say that in what way we can develop different kind of lead compounds. So we know that phytomedicine is a kind of representation of some therapeutically proven fraction or the active material which has some healing property. And we know that this healing process is not new concept, but it is existing since the advent of human civilization. So as and when the civilization is progress, as and when civilization goes on, this process of healing and process of various kind of therapeutics is also progressed with the help of the optionals which are available in the current era of the time. So as and when we develop the modern medication, a modern system of medication, a modern tool of medication, or novel tool and novel molecule for the medication purpose, we are also following different type of inputs from the natural kind of traditional medication system. How we and why it is important? Because see, we know that the phytomedicine, which is a kind of a vast era of biological activities and have been practiced not only in our country but worldwide. Various countries, they have different type of ancient medication system, and that is why, since it is thousands of years have already been proven for the upliftment of the individual health, so we can presume that the particular kind of the biological phenomena which is existing on the administration of this product may have different type of challenging kind of opportunities, which may be considered in terms of the solubility challenges, hydrophobicity, then low bioavailability, sometimes the toxicity kind of challenges. Altogether, they have different type of challenges which are associated with this product. Also, drug delivery technology have also attracted in enormous way, like novel drug delivery system can effectively transport different kind of phytomedicinal products, and which are capable of increasing the therapeutic index as well as the bioavailability of phytomedicines. So I mean to say, as and when we follow the traditional inputs or traditional medication inputs for the purpose of the modern medicament or modern therapeutical goals, then we have opportunity to develop these active fractions in terms of some novel drug delivery system, or in terms of some delivery system which are monitored with the modern science. Say for example, these days there is the concept of generation 3 phytomedicinal leads, which are very popular these days. G3 phytomedicine. G3 phytomedicine provide an ideal healthcare solution to people above the age of 40, as they can even be used for the long-term treatment of many ailments. But as and when they follow this phytomedicinal kind of products, and they use for the longer period of time, their effective effects or the lesser effects are being reported, and that is why we can consider that above the 40 age, the individual, they are also in need of some personalized kind of supplement, sometimes functional food, sometimes different type of complementing therapy with natural product, so that as and when they use this type of therapy for longer period of time, their different type of normal homeostasis is maintained. It means, suppose we are using some one molecule of synthetic nature, and if the dose is getting deviated incidentally or maybe because of ignorance, there are chances of the effect or undesirable effect on the kidney, on the other kind of biomarkers, and which may lead in disorder condition. And that is why a holistic approach is suggested to adopt for the maintenance of the health. Health is required to maintain its sustainability. As and when the health is maintained with the help of this natural products or the product of holistic potential, then definitely the different type of challenges or different type of disorders will not be the problem for the individuals, especially after the 40-year attaining of age of 40 years. So phytomedicines offer a huge opportunity not only for India but also for the world market for the herbal medicine, which is we know that it is being expected to reach 115 billion, and due to COVID, it was the export I will be showing you. The export is increased in multiple way, and it was expected, but beyond this, we have achieved it during last 2 years. So when we consider the huge scope of therapeutics as well as the international market, we know that different type of challenges are associated with our product. What are these challenges? We know that sometimes single target, single compound, or one disease, one target. It remains always the question of the researcher, question of the investigator. So with holistic approach of the therapeutics, the natural products, their isolation or the isolation of one particular component is having its complexity. But as and when we go for the holistic therapy, when we consider the synergistic kind of responses of the more than one compounds, there are different type of benefits associated. Just like multiple target with multiple active principles. So ultimately, our aim is to maintain the holistic maintenance of the body, and that is a kind of maintenance of normal homeostasis. So to achieve the normal homeostasis for the longer period of time, it means to achieve the sustainability of the life, we have to treat our body, we have to maintain our body in holistic way, and that is possible with natural product. As I told you, this natural product is having some inputs from different type of traditional medication system, different type of ethnobotanical inputs, different type of ethnomedicinal reports. So by using these ethnomedicinal reports and the ethnomedicinal inputs, with the help of some modern herbal medicinal practices, we may improve the overall therapeutic goals. Second challenge which is associated with this product is chemo variety, and this chemo variety is always remains one of the important domain of the researchers, where different type of unexplored or less explored kind of chemo varieties are studied, and ultimately these unexplored chemo varieties may have different type of synergistic responses, so which could be established with the help of modern analytical technique. So this one challenge, at one hand it is kind of complex and it is a kind of challenge, but at the same time this particular challenge may also remain as a opportunistic kind of tools. Then we know that the source and the quality of raw material which are variable. So it is also one of the challenge, but this variability of the raw material and the variability of the quality also having the opportunity to explore for the purpose of this medicament. Then sustainable growth and conservation of plant species. So as we know that India is having huge land, lush green kind of the area, which is most of the area is covered by forest land, and different medicinal species, they are continuously following up and downs in terms of their sustainable growth, in terms of their endangered kind of status. So to conserve this medicinal species throughout the countries, we have scope of working in terms of the sustainable growth of this medicinal flora. Then another challenge is herbal products are, we know that they are usually mixture of many constituents. So as we know that there are many constituents, our aim should also be they're targeting the various kind of constituents and their togetherness. In what way their togetherness is important for the holistic therapeutics as well as the overall safety of this particular product. Safety why? Because as we know that these medicinal products are being considered as a safe product. Why? Because they are not single moiety. They have different type of many constituents. They are a kind of mixtures of many constituents. So ultimately aim of this mixture is to complement the effect of each and every kind of constituent in terms of positive effect. Then it is also considered as a challenge, like one principle or one active molecule is unknown in many or in many most cases, and that is why for researchers to get the reference compound or sometimes to isolate a particular compound remains one of the challenge, and this challenge may also be considered as an opportunity for the researcher. Then selective analytical methods or reference compound not always available. So that is another way, another kind of challenge which remains before us to either we go for developing some fingerprint or some developing some novel method of isolation, and ultimately will be able to establish a reference compound. So it is one of the challenge, but it may be also a kind of opportunity. Then we have seen that during COVID era that many of the medicinal and aromatic plants were practiced with the inputs from the traditional knowledge and traditional system of medication. And during COVID, different type of group of therapeutics were established, and it's not new one, but it's a given in Ayurvedic and traditional medication system. Like common community is using different type of the immunostimulant and some kind of products of the herbal material for the maintenance of their normal homeostasis. So, and the persons or the people or the group of community who have used this particular normal procedure, normal products for the maintenance of their normal homeostasis, somehow they have faced, somehow they have maintained their immune system, and ultimately the COVID could not affect them at large. Then unexposed asymptomatic group was also there. So for that, in traditional medication system, different type of some functional foods like it is, or different type of some Ayurvedic preparations like Chyawanprash, Rasayana, Amrit Balataka, and another kind of Sanjeevani were proven very helpful during last 2-3 years. Then quarantine kind of group of community were also there, which were treated with different other approaches. Then mild COVID symptoms, the persons having very less kind of symptom or not, they are in danger zone, they were treated with different kind of product of Ayurveda and moderate to severe COVID symptoms. So I mean to say, during COVID era, we have witnessed that in what way the common community, where the hospital beds, where the medical facilities were not sufficient, they have maintained their overall health with the help of this natural product. Then we know that different type of medicinal and aromatic herbs which are used in Asian region, they are justified on the basis of the contribution of 2 countries worldwide. So we can say that the herbal kind of the medicament was established throughout the world or in various nook and corner of the world because of the contribution of 2 countries, that is India and China. Why? Because they have made use of plants not only for the cure of the ailments, but also for the functionality of plants for thousands of years. And that is why, according to WHO, which is functioning for health for all, and this health for all, we know that cannot be achieved without the preparations which is made up of herbal products. So herbal products are having different kind of avenues and opportunities to go for the different type of challenges which is related to health for all. According to WHO, we know that the market of the herbal products will be multiply, it is growing, and it will be 5 trillion by 2050, which indicates that the consumption, production, and the overall international trading of these herbal products will be increasing. And that is why it remains one of the, very sound opportunity for the researchers and or the herbalists or the scientists to look into the scope of the sustainable growth of this particular sector. Medicinal and aromatic plant sector in India has different kind of strength as well, as it is having abundant availability, since forest area is and the diversity is huge in our country. And that is why different type of the requirement, like different type of soil and the climate, then rich heritage of Ayurveda or other system of medication. Then we have a strong base of research and development laboratories throughout the countries. Skilled manpower, we have lower production, and the manpower cost is available in our country. So we can say that our well-developed pharmaceutical industrial scope is existing in our country, so that medicinal and aromatic plant products could be produced and exported in a large way, in a big way. The increasing importance of medicinal plant in fulfilling healthcare needs, particularly of developing countries, have also emphasized in various forms, like sustainable utilization of medicinal plants as per the TRIPS agreement, so that the different type of endangered species or the conservation of the medicinal plant always remains one of the challenge. And we know that in our country there are different 18 biosphere reserve which protect larger areas of natural habitat, and these habitats are maintained and monitored with the help of national parks and wildlife. Why I'm showing this? Because the conservation of medicinal plant, which is one of the challenge, is also the area of opportunity for researchers to look into the endangered species. And that is why this endangered species and the biodiversity can be studied with the help of these 18 biospheres. And we know that different type of national parks and sanctuaries are also functional in our country as per the Act of the Wildlife Protection. So this is the potential, like 45 plant species are there found in our country. Then 3,007 species of both higher and lower plant are of medicinal values. 1,000 medicinal plant species used by various contemporary industry. And we know that 80% are procured from wild area, mostly notified as a forest land. So only very limited, like less than 20% of medicinal plant are cultivated in field. It means our biodiversity is having its own importance for the purpose of the sustainable growth of medicinal plant. And the forest of Himachal Pradesh and Western Ghat are well-known area to supply a very large proportion of medicinal plant requirement of India, as well as the export from the India. So there are different type of opportunity which are based on the various challenges which I have discussed just now. So looking into the opportunities of these herbal preparations, we know that as and when we go for the opportunity, we have to look into global business angle. In what way the particular herb is having its global demand in international market. Then the next opportunity which we know that the quality with safety and efficacy issue. So as and when we develop the herbal material of the expected quality, we have to always monitor its safety and efficacy. Then in what way or up to what extent our drug is having phytoleads or the characterized phytoleads. Then what are the current therapeutic priorities which are available or which are the need of the hour. And of course, herbal products in veterinary purpose or animal healthcare. So these are the different type of the opportunities which are available with natural products and herbal medicines. Global business and research opportunities are having different type of segments, like it may be under the segment of phytopharmaceuticals, modern drugs, drug leads, or different type of intermediates. Then another area is preparations or herbal medicinal products, then traditional preparations or complementary medicines, some household remedies as well. So in this way, different type of businesses or the golden, global kind of opportunities are available for the research activities. In addition to these nutraceuticals, as I told you, function food, food supplement, and various herbal, beverages, they are also having potential to maintain the normal homeostasis. Then extract itself is one of the, very, famous opportunity for the researchers to develop either crude extract, semi-finished product, or the finished product in terms of the extract, and extract is being exported to various countries. Then aroma, essential oils, flavors, fragrance, perfumes, and the cosmetic product are also another area where global business and research opportunities are existing. Some pharmaceutical aids like gums, mucilage, edible dyes, sweeteners also remains as one of the opportunity for the global business and the research challenges for medicinal and aromatic plant. Then entomological preparations like insect repellents, insecticides, then some pesticides and the fungicides are also the area where this natural product and herbal preparations are having their potentials. So I mean to say the opportunities are huge. We have to decide in which particular area we have to go, and ultimately global business and research activities could be defined in a very strategic manner. As I was saying that the way of the herbal export value of Ayurvedic and other herbal products from India is increasing every year. Even though in the era of this COVID during 2019 and 2020, it was slowed little bit, but ultimately it has achieved its previous pace of the growth, and ultimately in the year 2022 it was again on its expected level of the export. So this is how this particular sector of herbal product is increasing. So how this increase and the sustainable growth is achieved? So when we talk about opportunity, or when we talk about the challenges, always we have to consider only two concepts of the herbal medicine. One is that is a requirement also, basic requirement of their drug herbal product is safety and effectivity. Out of these two requirements, safety comes first, and then therapeutic bioactivity. So keeping in mind the investigation for either for safety or the efficacy, we have to consider different type of secondary metabolites which are being present in our product, and that may be having different type of their synergistic type of potential as well. So the quality, when we talk about quality, the herbal products are majorly having two type of classes, like external classes, external kind of factors which includes different type of contaminations, may come from either metal contamination which is coming from external sources. Some pesticides may also come inside the herbal material, pesticidal residues. Some microbiome or microbes may also have their toxic kind of the concentration in the herbal material, like aflatoxin and many other microbiological kind of the materials, which may also automatically or during the cultivation or the collection process may enter in the mass of herbal product. So in addition to this, some intentional kind of adulteration or misidentification is also reported or also exists. So altogether we can say that these are some external factors. Drug itself is not having its own nature to get contaminated, but external factors, they are contaminating the drugs. So this is a group of external issues. Then another class of the quality, which is directly related to complexity and non-uniformity. So quality of the herbal is defined on the basis of the concentration and the quality of the secondary metabolite, and which may remain many times as a challenge because of the number of factors. Non-uniformity of ingredient in herbal product are the internal issues which actually affect the overall quality, and that is to be maintained with the help of real-time assessment of the quality checks in terms of the quantity determination of the secondary metabolites. So these are two classes. The one is externally, diminished or externally affected class of the quality, and another group of classes like complexity and uniformity because of n number of factors which is affecting the secondary metabolite. The quality may get affected at various places, as we know that when the herbal products are being cultivated or it is being produced at any site of the maybe forest land or the wild source, the product immediate after product collection or the harvesting the herbal material, either the material goes to some intermediate sites like dealers and suppliers, or directly it goes to market. From dealers, it is going to various processing factories, and from processing factories, it is processed herbal material is being launched in the market, or it goes to the pharmaceutical company. So this depiction is like at every time when the product is being transformed from one place to another, there are chances of the challenge of quality. From market, we know that some herbal products are directly reaches to the consumer, or it may be going to different pharmaceutical production houses. And again, from production houses, it comes to dealer, then to clinical hospitals, then to herbalist or practitioner, and ultimately it is reaching to the end users. So altogether, during this path, the overall quality is to be maintained, and that can be done with the help of real time or with the help of assessment of the packaged material or assessment of the various kind of degradation rate of the active materials which are present in the product. We know that there are different type of success stories as well, like different lead compounds have been developed by many scientists. Some of examples are enlisted here, like atropine, artemether. We know that malaria kind of challenges are associated, and now in a big way the malaria problem is meet out with the help of this artemisinin or artemether kind of moieties isolated from Artemisia annua. Atropine, colchicine, then different type of galantamine, then different type of digoxins, adiposide for the anti-tumor activity, morphine, which is we know that a very popular pain relief option, then pseudoephedrine, then vinblastine, vincliztin. See, overall investigation on this particular compound is, since they are now established phytoliths, they may be used as a lead compound, as a alone compound or a standalone kind of material. But as and when the material is used or prescribed in the form of extract or active fraction, even though that particular standardized extract is also capable enough to use for the particular therapeutics. So this particular the overall challenges and opportunities, how they are articulated. So articulation is done with the help of the standardization and quality control. We know that process of developing and agreeing upon some technical standard is in broader way the standardization. When we develop one standardization, or when the standardization process is monographed or the particular authority is approving it, it means this is a prescribed set of quality characteristics which must be present in a particular product. and eventually that product will be evaluated or will be regulated in terms of its quality check when the market is having this particular product, or when the market is the end user is collecting the particular product from the market. In that particular path of the travel of that product, the monitoring and regulatory check of the quality is done with the help of this set parameter of quality characteristic. So altogether, we can say that this is the agreement of the government, agreement of the production houses, and agreement of end user. And from end user, the government is putting some certain kind of norms. So altogether, we can say that standardization is nothing but kind of tool for quality process. Then we have our APSA attributes. These APSA attributes are nothing but authenticity, purity, safety, and assay. So in modern pharmacognosy, with the help of our different councils of the pharmacognosy, we know that APSA attributes are now practiced, and learning of APSA attributes are done with the help of modern methods like authenticity with the help of gross morphology, which may be the traditional or which may be the conventional, but the microscopy, chemical analysis, and DNA fingerprinting is most important kind of the process which is followed for the authenticity, by which we check the specific identity of a particular herb. Then purity, we know that no adulterant should be there, or adulterant is there, it must be under the particular regulatory norms. Then safety, we know that safety can be done with the help of the quantification of various kind of undesirable or harm effect. If undesirable and harmful effects are under the limit, then we say that this particular drug is passing this particular safety evaluation, and we may consider that particular product safe. So overall, this particular process is not the normal process, but in terms of research, we can develop different type of innovative models. Then assay is nothing but we know that chemical or biological profiling could be done with the help of chemical effects, and curative and therapeutic values could be articulated on the basis of assay procedures. So different type of these days innovative assay procedures are being developed with the help of the modern analytical techniques. Conventional data recording or conventional kind of the process by which we develop the data set of herbal products are like general data, identity description, and purity contamination. So we know that general data is nothing but they are evolved from geographical details where the particular drug is having favorable condition of its growth. Traditionally, we say that temperate or heavily raining area, forest area, or different other type of tropical or subtropical area. But in innovative way, we consider different type of biosynthetic pathway which directly affected by n number of factors which are available in those geographical area of favorable conditions. Then harvesting time like morning, evening, afternoon, or the time when the sunlight is it's a full of exposure to the plant, or the harvesting process like which particular process we adopt, like different modern techniques or traditional technique, and immediate after harvesting in what way we are processing the material. So all together this general data is giving us important inputs in terms of its quality. So if it is done with the innovative method of involvement during the collection of general data, definitely the quality of the particular herbal material could be devised in a big way. Then identity description like microscopic and macroscopic detailing, what kind of chemical profiling is done, then what is the TLC fingerprints with the help of densitogram or with the help of modern TLC or HPTLC. So altogether, identity description also remains one of the important tool for the identity description. In addition to that, molecular fingerprint, we know that metabolomics and these other image techniques or different type of system biological techniques are important for the purpose of the identity description of these herbal products. So we have to follow in our research strategies and research protocol to check whether the drug is really belonging to the expected species. So this could be done with the help of various modern kind of molecular fingerprints. Then purity and contaminants, although we can do it in conventional way, like foreign matters could be done in traditional procedures. But all together, when we develop the different purity and contamination parameter with innovative practices, innovative kind of processes, then definitely the reproducibility and the validated data will be helpful for the quality check, like foreign matters, ash values, then content of acceptable matter, water moisture content, then constituent of known therapeutic, we can say biomarkers, or some unknown kind of therapeutic activity, which may also again remain as a marker substance, but their concentration could be defined in a proper way. And also, in addition to that, if the innovative practices or innovative procedures are developed to find out the real-time pesticide concentration, heavy metal concentration, different type of microbiological purity or radioactivity kind of the parameters, then definitely will be able to achieving the goal of the opportunities which are evolved from the challenges. So in order to prove the constant composition of herbal preparation or this phytomedicines, adequate analytical methods always plays important role. And these methods involves like spectroscopic analysis, chromatographic techniques, DNA fingerprints, then chemometrics, then biopharmaceutical characterization, metabolomics, molecular imaging, and molecular mass imaging. So all together, when we involve these techniques for our research protocols or research strategies, definitely we will have different type of opportunities. So we know that DNA fingerprint is for the purpose of the identification, and we can develop different type of data sets, and that this data set is based on either this hybridization based kind of data set or PCR and sequencing based. So our aim is to develop the DNA data set that is specific to a particular plant species, which is helpful for the future reference of the taxonomic changes. DNA based kind of authentication also having its potential to control the overall international or widespread adulteration rate of a particular drug, because we know that the regulatory norms are getting very different type of diversity in when the product is travel from one country to another or one region to another. The range of the acceptance of various kind of toxins, various kind of aflatoxin, or different type of the minerals, metals, or other kind of undesirable limits are having their diversity, and that is why this DNA kind of based authentication is having its importance for the commercialization of product in various regions of the world. Need not to detail in hybridization technique, but it's having different type of clone and probe type of options, which we do. PCR involve various kinds of amplification of DNA sequences, and on the basis of sequences, we go for the confirmation of authenticity of the material which is supplied in terms of herbal product. Then chemometrics, what we have to do, we have to interpret the different type of three-dimensional data by using 2PCA, where different type of grouping is done and data set is correlated. Say for example, the analysis is done with the help of LCMS. So LCMS data set is hyphened, it is correlated with the other data which is coming from other chromatographic data set. So 3D and 2D data set is correlated with the help of principal component analysis, and ultimately it gives different type of variability in data sets, which is helpful for the authentication of drug material as well as the standardization of the product. Chemometrics, as and when this is a depiction how the analytical data is processed with the help of chemometric processing technique, where we get different type of chemometric fingerprints. After chemometric fingerprints, we extract and sort out the narrow range of data set. Ultimately, this narrow range of data set is useful for the marker compound analysis and herbal product fingerprint with multi-constituent determination. So ultimately, with the help of this interpretation, with the help of this chemometric interpretation, we are able to find out the marker compound of our particular therapeutic goal. So when we go for different type of profiling with the help of the system biological approaches, different type of metabolites, we know that the herbal drug is a kind of representation of human metabolome, which is containing some concentration of primary metabolites and the monitored concentration of secondary metabolites. So with the help of metabolomics, which is a kind of exhaustive profiling of metabolites, we are able to develop fingerprints of different primary as well as secondary metabolites. Here for therapeutics, we know that our target is secondary metabolite. So systematic study with unique fingerprints, which particular cellular process is leaving behind, is monitored with the help of metabolomics. What we have to do in metabolomics? Metabolomics is, as you know, it's a holistic approach. So plant to patient, starting with the plant side, in what way the product is being reaches or is being used by the patient or end user. That is the holistic kind of approach where we analyze the product for the safety and effectivity. So what we do in metabolomics, the plant organism is collected maybe from forest land or maybe from wild source or the cultivated site. Then sampling is done, and we know that in sampling we have different type of the metabolites, either primary like amino acid, different type of sugars, different type of acids or lipids, and some secondary metabolites like glycosides or some targeted kind of the phytolate, which is being developed in terms of it's a sample of metabolomics, and then it is subjected for derivatization. So derivatization is the requirement of the analytical instrument or analytical infrastructure which we have. So on the basis of this derivatization, our particular metabolites are able to be detected with the help of detectors in particular analytical procedure. So we follow under step 4 the analysis with the help of either LCMS, NMR or other kind of analytical technique, from where we get different type of data set, huge data set, and that data set is multiple in number and complexity in its nature. So we have to go for then conversion into narrow range of desired data set, and that is done with the help of data mining. So in data mining, we get only those signals or those spectra or those inputs which are having of our our therapeutic importance. So when we are able to finish our data mining, then this narrow range or the less number of the data set that is used for the functional elucidation of unknown gene function. So in this way, this overall process is starting from the number 1 living organism or plant material. It is reaching to different kind of feedback to the bioscience. So ultimately, metabolomics is giving us kind of the inputs for the worldwide registration, as well as registration means the regulatory norms fulfillment and acceptance of herbal product. That is a kind of so metabolomic profile it's a kind of potential key for the registration worldwide and the acceptance of the product. So we can say that the ability to provide scientific evidence in combination with quality control system based on bioactive ingredients, and this could be achieved with the help of metabolomic science. This is the representation of metabolomics or metabolome, where metabolome may be anything. As I told you, it's a bioprocess. As and when we administer any particular herbal product, either to animal or to clinical purpose human beings, then definitely the biofluid is having altered because of the involvement of different secondary metabolite. So the overall metabolome is nothing but it's a kind of biofluid, some metabolic footprints, some intracellular metabolites, or some tissues. So altogether, this particular metabolome is analyzed with the help of analytical technique like GCMS, LCMS, FTIR, then DIMS, that is direct injection kind of spectroscopy, or NMR. So majorly, this metabolomic investigation is done with the help of these analytical techniques. So when we have the oral metabolomic result of one phytomedicine, our aim is to integrate the oral findings. So this integration gives us different type of gene expression, different type of genomic data, genomic proteomic, then COVID-related kind of performance, various type of imaging or imagination type of the outputs, then clinical data like treatment, history, demographies, environmental factor, and all together. In addition to this integration of pattern of the what data set we receive, we are also able to go for the prediction of risk, prediction of the individual prognosis, diagnosis, what are the therapeutic responses in terms of device or drug, whatever, or environment, or any other type of normal homeostasis of the individuals. So prediction and risk is also possible with the help of innovative research, innovative involvement of the metabolomics. As I told you, that standardization of herbal medicinal product with ethnopharmacology and metabolomics is one of the these days multidisciplinary approach and well accepted approach for the drug discovery from natural source, where different type of knowledge-based drug discovery from natural sources are achieved. So we know that as and when we go for this natural product research, it is essential to distinguish two parts of developmental activities. So when we go for ethno kind of activities or ethno-medicinal kind of investigation, there are two things. One is field-based input or field-based studies, wherein local resources including functional and useful plant materials are collected. And another part is scientific study, which is being done in the laboratory condition, but that is based on the whatever claim is recorded from the local people or the local community. We have done one, one case study. I'm just in very short time, I will show you in what way we have done this. So this case study was like assessment of ethnomedicinal potential in selected region, where our aim was to micro-learning and multidimensional target based on sustainable socio-economic development process. So in what way this ethnomedicinal practice is having potentials in terms of micro-learning and multidimensional targets, which is not only for the treatment of the individual or the community of those rural community areas, but also in what way this ethno practices are contributing for socio-economic development. Then another objective was to identification of traditional key indicators and parameters responsible for growth and conservation of these useful botanicals. So we have conducted this particular study exactly at the center of the country. That is the one of the district of Madhya Pradesh known as Patil district. What we have done here, like we have considered survey area and its climate, then the protocol for the interview, for the recording of inputs, the plant material, its historical background, and the process of the collection and the storage of plant material, then the process of phytochemical profiling, how to develop the innovative profile. Then our target was to like pharmacological investigation, that is nothing but it was pharmacological screening, and the data set analysis, which was done with ICF, that is Informant Consensus Factor. So with the help of ICF, we have developed different type of relative importance of different plant species being used in those study area. Some correlation is already there. We can follow with the help of this formula, which is depicted here, and use values. So in this case study, we have collected the different data set, and the recording is done like this. I have just shown in one table, one slide, like although we have collected some 22 species. So the scientific name, where we have to give one voucher specimen of case study, what was the local name, what is the scientific family, then whether it is endangered, natural, or abundantly available, then what is the status, it is explored, unexplored, or it is endangered or less explored, like that. We have developed some code word. Then which particular plant is claimed by the rural community, then which method they have used for the purpose of therapeutics, and what are their recommendations. So, and what is the ultimate therapeutic value of that plant. So altogether, we have collected the data. This data set was developed in terms of different type of categories. So all the data collected, and we have developed this 29 therapeutic categories. So out of therapeutic categories, we have find out that a specific number of plants or plant species which are claimed in a particular area, and their different type of ethno-medicinal inputs were recorded. So out of this 29 categories, maximum 32 plant species were claimed to have therapeutic activity, and majorly on GIT. This gastrointestinal claims. 4 plant species were claimed for CNS function, and the project revealed that the promising and comparative memory enhancing properties of various herbs are available in the study area, and that can be explored for the modern pharmacological investigation. Under microlearning, it was observed that the time of collection and the harvesting method plays very important role for the functional importance of the plant. So I mean to say, if suppose we are collecting the material in the afternoon or the evening or the early morning, then their overall the medicinal value remains in different degrees, and that is why traditional people, they used to claim that the plant material should be collected in so and so time period. It was also found that many plant species have synergistic bioactivity, and these plant materials should be investigated in order to maintain the same. So instead of opting the isolation, opting of separation, we have to consider that the wholesome plant material is having its own importance. And also it was found that forest produce have more ethnomedicinal reputation than the other cultivars. So the material which is available in forest land, which is available in the area of the wild source, they have their own importance as compared to the cultivation site. Why? Because see, keeping in view whatever inputs I have discussed just now, we know that the modern science is also having its own kind of examples, which are very common. Like when we go for the fractionation of any active fraction, it's not necessarily that the fraction will produce the same compound, or the fraction of the active compound will not necessarily produce active compounds. Why? Because esterification of the acid groups on molecule, if alcohol solvent is used, then oxidation of phenolic compounds are also possible. So altogether, esterification and oxidation and the synergy is getting challenged, and it affects the overall fractionation process. So we know that the science, in modern science, this particular esterification, oxidation, and the synergy could not be ignored. Then most active compound found is not necessarily responsible for the major part of the effect. And say, for example, the senna, several dianthron glycosides are responsible for overall laxative effect. So in addition to that, in modern science, we also know that the phyto research is having its own multidimensional therapeutic goal. Like if we consider the example of wound healing, we know that wound healing is a mixture of various processes like cell proliferation, collagen formation, contraction of collagen lattice, and in addition to this wound healing is also impaired by microbial infection, destruction of cell and tissue by reactive oxygen species. So altogether, when we know that the one therapeutic process is made up of the multiple number of bioprocesses. So when you go for the isolated one, there are chances that the isolated compound may affect the cell proliferation, but not the collagen formation. So in this way, our target should be like we have to go for multidimensional therapeutics. Likewise, if we take the example of malaria, for malaria activity of natural product, the goal of new drug should be multidimensional. I mean to say, instead of having only killing of Plasmodium species, we must have some targets of anti-inflammatory activity or antipyretic activity, which is a common symptom of malaria. Because we know that the malaria patient is suffering a severe pain, body ache and all that. And also if the drug is not isolated, but it is developed in terms of active fraction, which is capable enough to address this multiple therapeutic goals, then the product will be having its own importance. Then we know that there are opportunities of the nature. We know that the medicinal plants and the phytomedicine, they are linking the doctrine of the signature. Like the plant, whatever shape plant is having, whatever color this plant is having, what are the color pigments are there in the plant, they have their own importance. So to start with any new plant material, we may also consider that this doctrine of signature or the signature or symptom which are linking these medicinal plant to the human health. So I mean to say, these are nothing but this doctrine of signature are some symptoms which are given by the nature to us to just correlate in what way this particular new species or this particular plant species have its own importance for the medicinal purposes. So nature has marked many things, or you can say everything, and created with different type of signature. Signature is nothing, it's kind of sign. So this picture shows that different food material is having different shape and sizes which is resembling with our body parts. So you might have seen that different parts like the one is like yellow and sweet, that is may have helpful for the spleen, red and bitter helpful for the heart, green and sour helpful for the liver, black and salty may have their activity for the lungs. Then carrot, when we cut the section of the carrot, it looks like the structure of or anatomy of the eye, and we know that nowadays modern science has proven that the carrot is very, very useful and beneficial for the maintenance of the eyesight. Then avocados, it looks like the infant looks like the cervix. So avocados, we know that it helps women balance hormone and unwanted kind of the birth weight and has protective action in cervical cancer. So why? Because earlier when the signature of doctrine was developed, it was presumed that this avocado may have some effect or some kind of bioactivity for the purpose of the maintenance of the cervix or the female. This is very good example of doctrine of signature, the walnut, which directly you can correlate that it looks like brain, and now many patents are awarded for the purpose of the investigation and findings on the brain function of walnut. This is the common example of kidney being sexually heals and help the maintenance as well as the normal homeostasis of kidney function. And we know that it exactly looks like human kidneys. Then tomatoes, we know that secondary metabolites, which is important not only as an antioxidant, but also it is having its functionality for the different type of function of heart or serious function. Then fix for the challenges related to the sperm count and the mortality. Then marine products are also having their importance for the function on the sperm and sperm problems. Here are some button mushroom. Button mushroom is, we know that button mushroom is very useful, and nowadays there are plenty of reports which improves the hearing capacities of the individuals, and we know that there are some secondary metabolites which are capable enough to improve the overall homeostasis of ear. Then grape seeds and extracts, they are very helpful for the function of the alveoli, function of the lungs cells, and also the many reports are there which was based on the COVID era, where the challenges of lungs, challenges of the function of this oxygen uptake and all that was reported. This particular extract kismish and different type of extract of grapes have proved their own importance for the functionality. Then the ginger, when we take different pieces, it looks like the stomach, and we know that ginger is very useful for the treatment of the GIT function, especially when the problem in stomach. Sweet potato, it is for the purpose of the pancreas, and we see it looks like the pancreas. So the glycemic index of the diabetic patients are maintained with the help of sweet potatoes. Olives assist the health and the function of ovaries. So this is a kind of different type of signature or doctrine of signatures which are existing throughout the world, throughout the nature, which is having their own importance. Ginseng for the purpose of stimulant and the relaxant. In addition to this, during COVID era, we have seen that hersingar, aloe vera, giloy, ashwagandha, turmeric, neem, red onion, tulsi, and black pepper. So on the basis of the inputs, on the basis of the maintenance of health during COVID era, we have observed that these are some 7 or 8 medicinal plants which are having their own importance for exploration. This could be explored for the purpose of the drug development for the COVID-19 problems. So to conclude, ethnomedicinal inputs plays very important role in quality control and externalization process. Modern evaluation and analytical procedures should be employed to explore and investigate traditional and ethnomedicinal experiences. I mean to say, we have to incorporate as much as possible this modern analytical techniques to investigate this year-old ethnomedicinal processes, to justify them in terms of their holistic potentials. Microlearning is the way by which microlearning is not only for herbal product, but everywhere in the life, every walk of life is based on microlearning. Sustainable target is achieved only through the microlearning. So microlearning in herbal research, again, it facilitates holistic approach of investigation. Quality attributes must be dynamic in terms of their quantum in the evaluation. As and when required, quality attributes should be modified in terms of innovative incorporation. Then externalization may be considered as an effective tool for the quality control. So as and when we go for quality control check, we have to set some standard procedures with reference to standard parameter, so that we are able to develop some effective and innovative tool for quality control. So altogether, this can be achieved by involving integration of both the things. One is modern and advanced analytical techniques. So this way, as and when we have different type of challenges, these challenges should be considered in terms of its opportunity. Then only the herbal products or the different type of natural product or herbal medicinal product could be developed in an innovative way. So this is how I have experienced many things which I have shared with you. If you have some question or queries, I will be happy to answer.
Thank you, sir.
I think I have taken extra time.
Thank you, sir. Thank you for a wonderful session. So if you have any queries in any contest, any participant, they can ask. They can unmute themselves and they can ask. You can ask your question on YouTube also. Okay. So, sir, I have a question from our one participant. That is, in recent herbal research, is it mandatory to isolate a single molecule, or more than one molecule together can provide a holistic approach for any specific pharmacological action?
So very good question. See, isolation is not mandatory always. If your research studies are based on only one moiety, then only you have to go for that. If your target is to develop some therapeutics, some therapeutic active or therapeutic formulation for the purpose of a particular therapeutic goals, then definitely need not to go for isolation. Your active fraction will be sufficient. Even active fraction will be having more importance than the isolated one. But isolation is, or isolated one is having its own importance in terms of its industrial importance, or in terms of its industrial reputation. If your target is to develop the most active medicament out of a particular herb, then definitely first of all you have to investigate in a holistic manner in terms of holistic development of particular active fraction, rather going to the isolated one.
Okay, thank you, sir. Any more question from any participants? Please mute yourself.
Participants are requested to mute yourself, please.
I think there is no more question from participants.
So I have also given my email ID. If
Okay.
anybody can fill, I will be happy
Okay,
to
sir.
have
Okay, sir.
their
It will be helpful.
any question, right?
Okay, okay, sir. Thank you. And sir, myself, Dr. Devjani Sarkar, Assistant Professor of JIS University, I would like to, on behalf of my department, Pharmaceutical Technology, I would like to, just a second. I would like to present you small token of appreciation, sir. Please
Thank you.
accept it.
Thank you.
Okay. Okay,
Thank you,
so
madam.
thank you once again, sir. And I will provide a link, feedback link for participants, and it will be open for 1 hour. Within 1 hour, you all have to submit your feedback. Okay. and you have to score 80% minimum for your certificate. Okay, thank you,
Okay,
sir.
thank you, madam. Thank you.
once
Can I
again,
leave?
sir.
Yeah, thank you. Thank you, you all.
Feedback link is provided on your chat box. Please submit your feedback within 1 hour.
আমার কাছে না এই ফিডব্যাক লিংকটা না বললে ওরা আর দেবে না সার্টিফিকেট। আর্সেনিকের আমার কাছে 12টা আছে, ওটা আমি আজকে টেনে দিয়েছি। ওরা টেনে দিয়েছে অলরেডি। হ্যাঁ হ্যাঁ
Okay, so thank you all the participants. So we are concluding our today's session, and we will continue tomorrow on provided time, 3 p.m. Okay, thank you all.